Okay, so Ella, I'll start by introducing you. So Dr. Ella Striem-Amit is an assistant professor at the Department of for Neuroscience in Georgetown University Medical Center and an Edwin and Elizabeth Richard von Masch, I hope I'm pronouncing this correctly, Distinguished Professorship in Neurological Diseases. She did her BSc in Life Sciences and Psychology at the Hebrew University, then an MSc in Brain and Behavior at the Hebrew University, where she already started to work on cortical plasticity and congenital blindness in humans under the supervision of Professor Udi Zohari. She then did her PhD in neurobiology under the supervision of Professor Amir Ahmedi, where she continued to investigate neuroplasticity in the blind and sensory substitution for vision. Her postdoc was done in Professor Alfonso Caramassa's lab, where she was funded by multiple prestigious awards. She now leads the Sensory and Motor Plasticity Laboratory at the Department for Neuroscience at Georgetown University Medical Center. Her research focuses on human adults with sensory deprivation, investigating how these unique conditions could further our understanding about typical brain and behavior, challenging the boundaries of neuroplasticity, cross-modal plasticity, and critical developmental periods. Her work attracts the public attention and is often covered by the media. She's active in disseminating science to the general public via multiple platforms. So Ella, we're very pleased to have you today with us to hear about your work. So let me welcome you to our seminar.
Thank you so much, Elan, both for inviting me and for that very generous introduction. I'll share my screen and hopefully there'll be no glitches. Again, as Sharon said, I'll be zooming through a few of my earlier years of work to focus today on visual cortex organization and individual differences in blindness. Since this is zooming and I can't necessarily see your faces, if you have questions, please speak up so I can hear it and address it on the spot. So as Sharon said, my lab currently focuses on congenital models of sensory and motor deprivation. And one of the things I want to acknowledge at the onset is that I work with extraordinarily small samples of individuals with congenital and full deficits. Although those samples are small, I believe they offer unique insight into some core questions about neuroscience, about the dependence on of the brain organization on sensory and motor experience, the potential and extent of plasticity possible in our brains, as well as the representational level that each brain region addresses across multiple senses. Now, with relation to the issue of small samples, I just want to point out that Micah Ramon and I are co-guest editing a special issue in Neuropsychologia, highlighting special populations and small samples and case studies. So if you work yourself on small samples and case studies, feel free to send your work our way. And if you are adamant against working on small cases and think this is the worst thing methodologically, also please submit something to this special issue so that we can discuss it. Okay, now practically what I'm going to be talking today about is what the brain and what the visual cortex does without vision. And that's something that I've dedicated the last maybe 12 years of my life to looking at. Trying to understand the balance between nature and nurture. We definitely know from seminal work, from Hubel and Wiesel's work, and from ongoing works, that visual experience carries a lot of effect on how the brain organizes, and that if you close a kitten's eye early in development, it generates long-lasting effects on the cortical organization. We also know that there's a lot of innate factors affecting brain organization, such that in monozygotic twins, there is more consistent organization of the visual cortex than in dizygotic twins. Now, I'm not going to be pitting it as nature versus nurture. We obviously know both things play a part, but those individuals with full deprivation, full visual deprivation, do allow us to study the role of experience and its role in plasticity. Now, I'll begin with works I did earlier in my career about the association visual cortex, and then move for the remainder of my talk, most of my talk, to talk about the earlier written topic, visual cortex. Now, with relation to the association visual cortex, we know a lot about its organization, right? We know there's two visual streams. There's the ventral what stream that carries information about what is out there in the world, objects, form, identity, and so forth. And there's the dorsal stream that carries information about where things are and how we may approach and grasp and use them. And in the ventral visual cortex, we know that there are different areas dedicated to different domains or categories of objects, such that there's an area that's dedicated to processing script, letters. There are areas processing faces, body shapes, scenes, and so forth. And one of the first things I started my career doing is trying to see whether the visual cortex organization for those large-scale organization types, the streams, the category selectivities, require visual experience. And over my PhD working with Amira Mehdi, what we did was try and map that out. We used sensory substitution to convey information to people born blind using sounds. And that allowed us to show that the large-scale stream division of labor of the ventral and dorsal streams can be found in the blind, and so can category selective regions, if we're talking about preference for script in the visual word form area, for body shapes in the extrastriate body area, and the overall connectivity pattern linking those regions to task selective and domain selective areas across the brain. Now, I'm zooming through this pretty quickly because by now it's already become pretty much a consensus about the association visual cortex in blindness. And there have been multiple studies showing that, for example, the area MT is selective for tactile and auditory motion in the blind in the same way that it's selective for visual motion in sighted people. And I can fill the screen with multiple examples of this phenomena. And there I did it. These are so many examples showing that overall it looks like the association visual cortex, and none of these specific examples matter per se. It's the general principle that the association visual cortex organization seems to not critically rely on visual experience or on visual features in the sense that it can process the same types of information through other sensory modalities. So the processing is more done for the same type of task. There is cross-modal plasticity. Yes, it's responding to other modalities than vision, but the processing is done for the same type of computation. And importantly, this organization is built upon the multisensory connectivity of those association cortices. There's actually very, very little change that we saw either in anatomical connectivity or even in functional connectivity of these regions in the blind. It's all based on how the brain is wired from birth to receive information from other sensory modalities and to link them to networks engaged in those types of behavior, in processing faces, in processing scenes and navigating through them and so forth. Now, over the last few years, there's been comparable findings in deafness as well, showing that not only in humans, areas that are dedicated to processing auditory rhythms are processing now visual rhythms in people born deaf, but also in beautiful works by Steve Lumber's group in congenitally deaf white cats, that the association auditory cortex also seems to have that type of task-based or computation-based organization, suggesting that its roles are really in calculations that extend across the senses. Now, what I've been doing in the last few years of my life is trying to extend that task preference and the innate type of organization for calculations to the action and motor domain, working with people born without hands who act with their feet, to show that not only is the association area association cortex engaged in sensory calculations that are independent from the input modalities, but it's also independent of the output way of manifesting those behaviors. So action observation, tool observation, and even action execution can be found in individuals born without hands in the same typical areas engaged in hand action. And one main line of research that my lab is currently engaged with is trying to see how that extends to the actual motor domain, to the motor association cortices, and to see whether motor hand areas are actually engaged in action type preferences, for example, for reaching, for grasping, and so forth with the foot when the hands are missing. Now, that is a gist of summarizing 12 of my last research work into 12 minutes, generally showing that the association cortex plasticity pattern is based on existing connectivity. It's based on accomplishing the same functional roles and the same behavioral outputs. And that pretty much summarizes what I have to say about the association visual cortices in the blind and association cortices in general. But what is going on in the primary visual cortex, right? My only answer at this point is it's really complicated. And there's been at least a couple of decades of research now diving into this and just showing how complex and how non-trivial the answers to this question is. Now, for my part, I've done the almost trivial thing, which is to see whether early visual cortex responds in a retinotopic way in people born blind. Now, since I couldn't stimulate the primary visual cortex retinotopically in those individuals, and in truth, trying to stimulate and doing a comparable analysis to retinotopy did not work in the blind, and I'll get to that soon. What we tried doing first was looking whether the networks are still there at all, right? Because I've kept stressing how the roles of innate network organization, and by innate, I just mean present at birth. I'm not saying anything necessarily about the genes driving these behaviors, though I can get to that later. But what we did was try to see whether a retinotopic type organization can be found in the networks of people born blind. And by sampling functional connectivity, that is sampling the time course in different parts of the brain, specifically in different segments based on retinotopic organization in people born blind, we were able to show that the main principles of retinotopic organization for eccentricity, for laterality, and for elevation are found in the blind brain, showing that at least... Yep.
Can I just ask a question?
Of course.
When you say they're born blind, what exactly, I mean, do they have any vision at all? If they have any vision at all, you could get those major pathways from their visual input.
I completely agree with you. I didn't add those slides to this talk, but in this work, this is a group, a larger group that has all types of blindness etiologies, including people who have eyes, were born with eyes, and have lost them due to ROP. So we can't exclude in the majority of those individuals whether this had to do with retinal waves in utero. We did have a subsample of microphthalmia individuals who did not develop intact retinas. Now, none of them, to the best of my knowledge, had no retinas, but even in those individuals, we could replicate that finding. So I dare say that this does actually extend beyond the effect of retinal waves. Does that answer the question?
Yes, thank you.
Thank you. Anyway, that just tells us that the networks are there. It doesn't tell us what these regions do. And what people have shown that these regions do is quite remote from visual processing. And over a couple of decades now, it's been shown repeatedly that the early visual cortex can actually respond to language in people born blind. And you see 3 different examples from 3 different groups showing the involvement of the blind early visual cortex in verb generation, verbal memory, again, verb generation here, and syntactic processing in people born blind. And that has brought about multiple hypotheses about how that type of takeover could occur in the primary visual cortex of people born blind, essentially suggesting that while in the sighted brain, the visual cortex, including the early visual cortex, receives information from not only visual input, but also information from auditory, somatosensory, and higher cognitive regions, in the blind, it may receive information from all these modalities. And that specifically in the primary visual cortex, the takeover is done based on higher cognitive regions, such as the frontal cortex, taking advantage of this empty piece of cortex in order to drive it to something completely different. Now, essentially, that means that it was literally suggested that the visual cortex is functionally pluripotent. It can organize to whatever it wants, essentially, even to things that are completely remote from its original organization. And to me personally, having seen so many research lines and addressing and supporting the role of connectivity in driving brain organization, and seeing great limitations of plasticity in other models of deprivation, it made little sense, in truth. I found it, I've seen the evidence myself. When I was working with Udi Zahari in my master's, I firsthand collected data sets showing activation for verbal memory in V1. But what really bothered me is that I couldn't find a way, a mechanism allowing that level of plasticity in the primary visual cortex. The only way I could think that this could come about is if cortex wasn't pluripotent, but if the early visual cortex would actually be responsive to language in the sighted brain as well, such that both the connectivity pattern and to some degree relevant information and function would be arriving at the primary visual cortex in people who are sighted. And having postponed quite a few years from diving into that rabbit hole, I finally dove in and tested that with a really robust language comprehension test, looking at forward versus backward speech of simple sentences. The advantage of backward speech is that it maintains the auditory envelope and a lot of the auditory low-level features, while depriving, making that stimulation without semantic content, without language content. And to my great surprise, honestly, I found that not only were left lateralized language areas in the frontal lobe and the temporal lobe activated for language processing and language comprehension in sighted people, we also found a very, very strong and very consistent activation in the calcarine sulcus and specifically the ventral visual left lateralized cortex in people who are sighted. Just for people who aren't used to looking at brain slices, I'll briefly go through this. This would be, can everyone see my mouse? So I'll be presenting a lot of brain images.
Yes, yes, your mouse is seen.
Thank you. So this is the left hemisphere, this is the right hemisphere. These are the lateral views and medial views. The central sulcus is over here, the lateral sulcus is over here, and this is the calcarine sulcus. So over here, you're seeing essentially early visual cortex activation. Now, I'm not saying that V1 is as activated for language inside the people as it would be for, say, vision, right? The signal in this area is way, way, way weaker for processing language, but it is way stronger for speech than for reverse speech. And the hemodynamic response function looks typical. It doesn't look completely messed up. Now, obviously, the first thing we thought of was, well, it has to be imagery, right? If we're talking about visual recruitment. And in this case, we're also talking about concrete sentences, things like the large gray animal is an elephant. So people could technically imagine that information, which would result in visual cortex recruitment. However, to the best of my knowledge, visual imagery results in a very different spatial profile of recruitment of the visual cortex. First of all, it recruits way more of the association cortices than the primary visual cortex, unless people are really imagining detail of the visual scene. And also it was left lateralized. It was way stronger in the left V1 than in right V1, and in V1 as compared to V2. So it really didn't match what we would expect from imagery. Now, in truth, that's not a complete answer. The only way you could address that question of imagery was to test whether the same areas would be recruited when we're talking about abstract concepts. So that's what we did. We looked into responses of the primary visual cortex, and you can see it over here, responsive, when people heard abstract words. And we were able to show that not only is it responsive for abstract words, but it's also as responsive for abstract words as it is for concrete object names in sighted people. Overall, this suggests that V1 is responsive to language in the sighted brain. The way that I can explain it, and it's not a complete explanation, is that it would perhaps be a part of a predictive coding scheme to guide perception based on input from higher level regions. In truth, I do not have a conclusive explanation for that. But for my purposes of explaining the plasticity observed in the blind brain, it really is reassuring because it really shows that V1 plasticity, even in blindness, can still be accounted for with reweighing of existing inputs and existing functions in the sighted brain.
Ella,
yeah.
Sorry, it's Marlene Berman speaking, sorry. My question is whether you know that this activation in visual cortex in language plays a functional role. So if there was a lesion, are these people going to become aphasic?
First of all, it's a great question. The answer is that I don't have an answer, but I wouldn't at all assume that that's the rule. I would assume that it plays a functional role. Otherwise, metabolically, it shouldn't be there, right? The brain has evolved to save energy. So I'm kind of assuming that most connectivity patterns that we would find, most activation patterns would have some functional role. But obviously I wouldn't expect it to be anywhere near as relevant as to cause aphasia. Even in people born blind, there's very little evidence. There's indeed, I think, one TMS study showing that TMSing early visual cortex results in supposedly semantic errors in people born blind. So even there, I think I don't assume that at all.
I think it's hard to know. I mean, this could just be the result, not from a metabolic point of view, but this could just be the natural cascade of signals that are both feed forward and feedback as a function of the underlying connectivity. But it's really epiphenomenal.
It completely can be. I completely agree with that at this point, and in truth, just based on the little I know about the intact visual cortex, I wouldn't expect that to play a very strong role either in visual perception and definitely not in language processing. It could be completely epiphenomenal. But again, for my purposes of trying to explain what we see in the blind, at least it gives us an anchor into how what we see in the blind could even come about. And okay, that was me trying to slow myself down so that I speak concretely. Okay, so to me, that would have answered the question, except that the language isn't the only thing activating the early visual cortex in blindness. I could fill a screen, again, with images of studies showing early visual cortex activation, and in some cases, by the way, also effects of TMS for touch, audition, spatial attention, memory, math. I'll spare you the array of pictures on the screen. But that really still, to me, made it unclear whether there's even organization in the early visual cortex, or whether it's just a cortex that's up for grab. Now, each of those activation patterns can in itself be justified by existing connectivity to the visual cortex. Even early visual cortex, even primary visual cortex does receive information from other senses, from feedback connectivity, for sure. But I was wondering whether there's functional specialization in the blind early visual cortex. And then I realized that what I was actually asking was whether there's consistent specialization in the blind visual cortex. And that's not a trivial assumption. Plasticity in the blind so far has been mostly characterized at the group level only. And that is a disadvantage of studying small cohorts, because you cannot really try to show pattern or to explain patterns that are only seen in some of the participants. But that made me think whether those contradiction or conflicting results about what early visual cortex does in blindness could actually arise from intersubject variability. Under the assumption that the blind visual cortex in general may receive information from all those senses and all those higher level cognitive functions, but it can organize differently in different individuals. And more broadly, that made me think of the question of what is it that makes us assume that brain plasticity is even consistent? Or rather, what is the role of experience in generating consistent brain structures and function for the visual cortex? Now, what we do know about brain variability, and here I'm switching gears to talk essentially only about functional connectivity patterns, because that is a great way of getting a larger sample of individuals to do the same thing. Broadly, individual differences are really highest in late developing cortex. You can see here all those areas marked in orange and yellow. The frontal lobe, the parietal lobe, the temporal lobe, the default mode network, whereas the visual cortex itself, both in terms of connectivity and also in terms of functional recruitment, as we've seen in multiple studies over the years, is very, very consistent across individuals. Now, late developing cortex has had more time to be affected by variable life experiences, and that brought other researchers to speculate that there could be a link between variability and experience. But to our purposes, I wanted to use the blind cohort to test whether visual experience similarly shapes the networks across people. And when we think about visual experience, we're less thinking about divergent experiences and more thinking about a very consistent type of stimulation with specific visual regularities and statistics of the visual scene that can actually be expected to decrease variability between individuals. So I just used a very straightforward question to test whether visual cortex would be less consistent in the absence of such visual experience. I took resting state functional connectivity from V1 in a large cohort of 25 congenitally fully blind adults and 31 sighted individuals, which I gathered with Amir Ahmedi and Yan Chao Bi throughout my career. And I want to explicitly thank them for allowing me to use that for ongoing research, and tested whether the functional connectivity across the brain would be similar or different in its variance between the blind and the sighted. And what we found is that there's a huge difference across multiple parts of the brain in the variance between the groups. And that difference was almost entirely, with the exception of one tiny clusters in the right auditory cortex, almost entirely blind individuals showed more variable functional connectivity between their visual cortex and essentially almost anywhere else in the brain than sighted individuals, suggesting overall that visual experience contributes to brain consistency across individuals. Now, my question to begin with was about whether plasticity results in more variability, right? Whether the blind would show more variable patterns in areas that are reorganized as a result of blindness. So what I did was to try and see whether areas that show increased variability are correlated to areas that show reorganization in blindness or plasticity, areas that are changed in their functional connectivity between the blind and the sighted. And overall, the answer has been a resounding yes. The two spatial patterns are very strongly spatially correlated. You can see that both to some degree visually, but moreover, when you compare that to scrambling the voxels. And it's unique to the visual cortex. If I run through this entire procedure again and again and again for other Brodmann areas in the brain, not for Brodmann area 17, then the correlation between plasticity and variability goes down to zero. And most importantly, areas that we have a stake in, areas that show plasticity in connectivity and in function to the primary visual cortex, such as the frontal left lateralized cortex, have way larger variability in the blind in their connectivity to V1 than in the sighted. Overall, suggesting that plasticity in blindness generates more variable outcomes. Was that in any way clear? Or are there questions?
Well, it seems from your map that there was a very strong variability in the left frontal cortex. Even looks equally to early visual cortex, and yet you say that there is no plasticity in there. So
I'm not saying that there's no plasticity. Of course not.
So I misunderstood it. I thought it was unique to V1. The correlation to the plasticity effect.
My apologies. Then I must have not been clear enough. The entire early visual cortex is considered to be plastically reorganized. I'm focusing on V1 for both methodological reasons, to look into functional connectivity from a single seed in that sense, and because V1, its organization for language is maybe just to me, but seems to be more strange or more drastic in terms of its reorganization. The earlier the visual cortex, the more we assume its functional role would be in retinotopic mapping of the visual space, and the more I was surprised personally that it's recruited for other tasks and computations.
Can I just ask, does the variability correlate with the extent of residual vision in your blind patients?
Again, great question. I have not tested that in terms of correlating them, but I'll show you in a second that it doesn't necessarily correlate to vision, visual or blindness etiology and other parameters.
Thank you.
All those blind individuals have no ability to localize light. So the most visual abilities they have is perceiving whether it's light or dark outside.
Okay, thank you. Thank you.
Thank you. So the plasticity is variable, but what are those outcomes looking like? What are the patterns of plasticity that we can observe in blind individuals? And again, what I'd predict is that in some blind individuals, V1 would be more functionally connected to maybe left lateralized, but maybe overall frontal regions. And in some individuals, it would be more connected to sensory, somatosensory and auditory cortices. And essentially that's what we find. This is an extraordinarily preliminary attempt at looking and parsing out using hierarchical modeling, hierarchical clustering, different maps of individuals. But it does show that there are very distinct patterns of plasticity, such that some individuals have positive, really strong functional connectivity between their visual cortex and sensory motor cortex, whereas others show negative correlation to this area. Other individuals are characterized by really strong auditory functional connectivity, and other individuals, which is in fact a relatively large group, has really strong functional connectivity to language areas and to inferior frontal cortex in general, without increased or largely increased functional connectivity to sensory motor cortices. Now, with relation to your question, of course, one of the first things we thought of was whether it just reflects something about the variability of reasons for the blindness, either the blindness etiology, if it's genetic, or the blindness, the visual capacities. And while I haven't don't have statistics to show for this yet. What you see here is different causes of blindness, such as ROP and microphthalmia, as being pretty much dispersed across the entire range of individuals and clades. And more specifically, those two participants both in clade 3, but in different parts of it, are sisters, suffering from blindness from the same genetic cause of microphthalmia. And they end up with quite dissimilar patterns of plasticity and functional connectivity to the primary visual cortex. The next thing, of course, is to see whether this is due just to the shared experiences of the sighted, to the absence of visual experience, or whether we can actually pinpoint some specific compensatory experiences of the blind that can explain the variability in functional connectivity and brain reorganization. Now, at this point, I don't have a lot of behavioral data collected from this entire sample of individuals, but I do have their degree of schooling. And since language and executive functions are explicitly trained through years in the education systems, I assume that it could affect reorganization differently among these individuals, and it may actually contribute to connecting the visual cortex more to areas that are involved in schooling. And that's actually what we found. We found that in individuals with longer duration of education, being just in formal education, primary visual cortex is more strongly correlated or functionally connected to the left inferior frontal cortex, suggesting overall that plasticity in blindness generates more variable outcomes that are affected both by visual experience and by the experiences of the blind individuals themselves. Now, in the future, I'd like to go into the direction of trying to see whether those individual differences can be useful for tailoring rehabilitation and assistive technology for individual people suffering from visual loss. And obviously, extraordinary claims require extraordinary evidence for those types of, for this to be convincing. But let me just show you one data point suggesting that this could actually be the case. In the last couple of years, I've been collaborating with Pavan Sinha from MIT, working on this incredibly beautiful project called Project Prakash, or Project Light, in which they perform sight restoration operations on kids and young adolescents from rural India who have been born with dense and bilateral cataracts and who grew up essentially blind. They perform these operations, and then they track the visual development or the visual capacities of these individuals as they regain sight. Now, I'd like to put the regained sight in quotations marked to some degree, because sight restoration following long-term and early onset blindness is very non-trivial, and it results in very incomplete visual capacities, such as problems in parsing the visual scenes and identifying object borders. And there's also a large and very unexplained variability in the outcomes, both across individuals within each cohort, but even across cohorts in different countries, performed either in Ethiopia or in different places in India as well. And I was wondering whether brain connectivity and or its change following sight restoration can explain the differences between individuals in their visual capacities. Now, first of all, of course, we need to test whether there's even change in visual cortex connectivity following long-term blindness. So I took advantage of the fact that these individuals, or at least some of those individuals, undergo functional MRI both before their operation, at different time points after their operations for both eyes, and in follow-up visits to the clinic and to the hospital after the sight has been restored. And today I'll just show you a very straightforward comparison between the pre-op scans and the follow-up scans following a month and a half or even 6 months after the operations. Again, I have a small cohort here. These are 10 individuals for which I have both pre-op and follow-up data. But since this is within individual comparisons, I find that I can make some limited claims based on that data as well. So what I did was take the data generously contributed by Project Prakash and its people and use resting state functional connectivity from the left calcarine sulcus on an individual basis to compare functional connectivity between after the surgery and before the surgery within individuals. And what you can see here is that even following cataract removal at different ages in life following early childhood, the primary visual cortex can change its functional connectivity and becomes more functionally connected to the bilateral pulvinar and to the dorsal stream, specifically dorsal V2 and dorsal V6. I'll focus now on V2 because this area had very consistent increase in connectivity across the individuals, such that I think 9 out of those 10 individuals showed functional connectivity increase to that region. And basically this slide just tries to show you that there are functional changes following sight restoration. And much like the behavioral improvement in those individuals, at least at that stage of learning to see, more behavioral improvement in seeing for dorsal stream functions than for ventral stream functions. And that's paralleled in increased connectivity to the dorsal rather than ventral stream.
Ella, can I ask about I want to ask the top views of the cortical hemispheres. Are these the patches? Do they show functional connectivity to V1 seed before, or do they show the difference between the after and before? What do they show?
Sorry, I'm sorry if I was unclear. These maps just show the difference in functional connectivity within individuals between the follow-up scan and the pre-op scan. I didn't test whether they were strongly functionally connected to V1 before the operation. I can check that.
Okay. Thank you.
Of course. But for our purposes today, what I cared about was to see whether functional connectivity change is correlated to acuity improvements, to the resulting vision from sight restoration. And again, today I'm only showing information about changes in acuity, and we have additional data, they have additional data to share about other visual tasks that are more engaged in functional vision per se. But what you see here are areas in which acuity improvement, so the difference in acuity between those two scans correlated to the functional connectivity change to the primary visual cortex between those two scans. And again, tiny cluster, very small sample, but it looks like resting state functional connectivity and acuity changes are correlated in dorsal V2 again. And that area seems to be increased in connectivity. This is just an overlay of the previous map of overall increase in functional connectivity and areas that show variable increase in functional connectivity that's related to acuity. So V1 generally seems to increase its connectivity to V2, to dorsal V2 across all those individuals. But the more it increases its connectivity, the more acuity improves in those individuals. Suggesting that the visual connectivity is functionally irrelevant to the quality of gained sight. Now, last, and again, I'll sprinkle grains of salt about it in a second. I literally have images of grains of salt. but I wanted to test if functional connectivity before the operation can allow us to predict acuity improvement in those individuals after the operation. So I correlated the functional connectivity from V1 pre-op, just that time point, to the acuity improvement, the gained acuity difference between the follow-up and pre-op scans. Bear in mind that acuity improvement is not correlated to the acuity at the pre-op stage. And what I found, in truth, to my great surprise, was the recruitment and the correlation to the entire ventral attention network, including the inferior frontal nodes, the TPJ node, and the posterior STS nodes of the ventral attention network to be strongly correlated to the gained acuity in these individuals, to the degree that, again, 10 individuals sprinkling grains of salt, but I could actually train a linear regression model based on the data, the functional connectivity from V1 to TPJ, and predict the outcomes in terms of acuity gains. I've just sort of pre-registered this correlation so that we can test whether that occurs in an out of sample next individuals come through this pipeline. But it does suggest that preoperative resting state measures may actually allow us to predict who would most benefit from cataract removal, and or what networks are involved in improved sight recovery in these individuals.
So do you think this has to do with their personality in a way? I mean,
yeah, I don't know. So one of the things, well, it's a grant I wrote, let's hope it gets funded, but one of the things I want to collect a lot more behavioral data about individuals, both in the congenitally blind cohorts and in these individuals, so that we can try and make sense of what is it that allows for good sight restoration. And in truth, I don't think acuity is the right measure to be testing. True. Acuity isn't really necessarily correlated to how well they perform visually in everyday life. Project Prakash Pavan and his team are collecting multiple behavioral data points from multiple behavioral experiments. So I hope that in the future, and again, with a larger sample and pre-registering those predictions, we would be able to make some sense and see who gains from this approach of sight restoration as opposed to other maybe different assistive technology approaches.
Does it correlate with both the age at which they had the cataract removed and the extent of the cataract at that stage?
Again, great question. It does not correlate. None of the findings I've shown here correlates to the age of cataract removal. In all these individuals, there is evidence in terms of nystagmus and so forth that they have had cataracts from a very early onset. And again, we're basing it to some degree on family reports that the cataracts were dense and that the kids were behaving as blind very early on. But then nystagmus itself is supportive evidence as well, that this is really early onset and relatively full deprivation. And again, these findings are not correlated to the acuity pre-op either.
They're bilateral cataracts, bilateral
dense cataracts, yes. To conclude what I've shown you today, I hope I've convinced you that congenital blindness results in functional plasticity of the visual cortex. That has been shown by many, many people for decades now. We believe now that the association cortices retain the same function for other senses, not only in terms of vision, but also in the deaf and in people born without hands. That the association cortices are just abstracted. Their processing and their representation level is abstracted from those low-level features. And that even early visual cortex can functionally change. And again, I'm not limiting this to V1, but even changes to remote functions are still based on existing functions and existing inputs found in the typical brain. And the cortex isn't completely malleable. Now, plasticity in blindness does generate more variable connectivity suggesting that sensory experience plays a role in affecting individual differences, and it can even change following sight restoration, even after long-lasting and early onset blindness. Together, those two last points suggest that individual differences in brain connectivity may be informative to predict treatment outcomes for ascribing individually tailored assistive strategies or even restorative approaches. And in the future, I'd really like, as I said, to correlate to more compensatory capacities in the blind and to additional measures of sight restorations over a longer duration of recovery. And also to expand that level of that approach of looking into individual differences to deafness, where we have in the past seen that there is spatial correlation, there is functional connectivity in the deaf auditory cortex that is linked to tonotopic-like features, but that connectivity and that topographic typicality is really variable between individuals, such that maybe the variability, either in intact function or in functional connectivity to other cortices, more broadly can tell us something about what are the best approaches to tailor to individuals suffering from sensory loss. I have to advertise at this point that I'm looking for passionate postdocs for multiple lines of research in my lab, both for the action execution work with people born without hands and for the individual differences line of research. So if you're interested, please talk to me. And I'd like to thank essentially everyone I've worked with over the last 15 years, starting with Amir, work with whom I've shown at the beginning of the talk, and also for allowing me to use resting state data we collected together. Smadi and Arno, who worked with me on the functional connectivity origin topic maps. Yan Xiaobi and Xiaoying Wang, who helped me collect the functional connectivity data from the other cohort of blind. Anna and Alyssa, with whom I'm working on the language recruitment. And of course, Pavan and the entire Prakash team, including Tapan, Sharon, and Shlomit, who generated this incredible data and shared it with me. And of course, my slowly growing lab, and especially Shriparna, who helped me analyze the individual differences data. And thank you very much for your attention.
Thank you very much, Ella. And I'd like to invite everybody to give a big applause to Ella for a wonderful and inspiring talk. Do you want to leave your screen shared, or maybe?
Oh, as you wish.
No, it's okay. Okay, so I'll move to
I thought I'd see everyone.
Yeah, that's an idea. Yeah, definitely.
If there are specific questions, I'd be happy to address them with slides, but are there specific questions? I can answer.
I have a question. Your functional data you started with was all about the language function in V1, but then when you went on to the later data, it was patterns of resting state connectivity that were variable. And I wonder how far these might reflect takeover of V1 by different cognitive or sensory functions in different individuals. That is, are some people using V1 for language, other people using V1 for blind navigation, or and that kind of thing?
So that's essentially where I would like to continue looking into this. So I'm fingers crossed that my grant gets funded so I can acquire exactly that data of collecting not only functional connectivity data and behavioral data, but also functional recruitment data to see how that correlates to variable recruitment between individuals.
I mean, how much, is there any evidence on how much variability there is among the congenitally blind in their in different behavioral and cognitive functions? That is, do some blind people get very good at Braille, and other blind people get very good at navigation? And is there these kinds of differences?
Certainly, there are certainly differences in different compensatory strategies that individuals use. And some of those capacities are also correlated to the degree of functional recruitment of the visual cortex for those different abilities. What has been less studies are the spatial patterns of this, and whether in individuals where, say, auditory recruitment is lower, this area is actually being taken over by, say, touch. So each of those data points was collected separately, so that it's very hard to compare whether the individuals who have lower engagement and lower functional connectivity for touch in primary visual cortex have a heightened recruitment for audition and so forth.
Marlene has a question, or is that okay?
Yeah, thanks. Thanks, Ella. That was really, really interesting. My question has to do with what kinds of formal principles we might be able to extract from the findings. So, for example, sometimes we do see a pattern in the blind that makes us think that this must be nature. And sometimes we see a pattern that makes us think, well, this is experience dependent, so it must be nurture. Sometimes we see a pattern like the activation of the ventral attention network. Why the ventral attention network? And then there is this variability. So I guess I'm trying to sort of step back and understand whether kind of a priori we have some kind of mechanistic framework that we can apply that allows us, I mean, this is a hard question, to kind of understand the constellation of the data rather than just following the trail of whatever the next empirical result is. So I'm sort of struggling to understand that framework.
Fair
Thanks.
enough. If I understand your question, you're asking, where is that theoretically leading us? And I agree with you that is always at the back of my mind. The reason I started looking into this in recent years is because I couldn't make mechanistic sense of what I was seeing in the blind visual cortex. I greatly believe, and I think that at this point is based on a lot of data, that the brain and specifically parts of cortex can only reorganize to some degree and to things that they have wiring to do, because there's very little evidence there's anatomical connectivity differences. So what I'm trying to do is parse out how the intact connectivity of the sighted brain with minimal modifications can account for what we see in the blind. And I don't have a full answer to the nature nurture weights question. I don't expect to have one anytime soon. I think to a large degree, the anatomical connectivity is already present at birth. And so all the modifications possible based on sensory experience are very limited and are more in terms of degeneration of visual tracts and in terms of changes of weights to existing anatomical connectivity. And in the smaller scale fine-tuning of the networks in terms of inhibition excitation balances and in terms of fine-tunings of those maps, which we know occurs postnatally and we know occur based to some degree on experience. Now, how do I explain those variability findings with relation to that? I still don't know. I think at this point, although this is theoretically driven, I need more data to make any meaningful conclusions.
Thanks.
Thank you.
I have a question. Hi, Ella. Wonderful talk. Thank you very much. Very inspiring data. You showed this correlation between plasticity and variability, if I understood correctly, and it was particularly strong in V1 or in early visual cortex. So how does this relate to this idea of early visual cortex being the pluripotent cortex? I didn't quite understand to what extent this data speaks for V1 being pluripotent or against it, or what your stand is on this.
Okay, I'm sorry if I wasn't clear on that. I was trying to make those 2 points separate enough, but I apparently wasn't. I don't know what it could mean that a cortex would be pluripotent. I personally don't think that's possible. At least maybe I don't have strong enough imagination to find a mechanism that allows it. The link between connectivity, functional connectivity, and variability and plasticity has to do with taking the existing connectivity in the sighted brain and changing its weight slightly. What I'm not showing in those slides is an additional analysis that I have that suggests that areas that show plasticity in the blind are areas that are also quite variable among the sighted themselves. So it's connectivity that can be found in the sighted. It's more variable in the sighted as compared to other regions. And it becomes even more variable in the blind, allowing recruitment of the visual cortex to different things. And again, saying that recruitment is functional again, and that V1 is a functional component of those networks, and that if you TMS it, it will result in language deficits, auditory deficits, or tactile deficits, is something that there is some evidence for, but I would like to see stronger evidence, let's put it that way.
Thank you.
Thank you.
Oliver, do you want to unmute yourself?
Thank you. I'll do something more than wave my hands then. Presumably, we have to ask, what are these pathways connecting V1 to the rest of the brain doing in typical development? And one presumes in the sighted, one of their important roles is in the modulation of vision through attention that can be directed by various exogenous factors. And if that's so, I wonder whether it provides a route for thinking about how these differences arise among different blind people and how far they might be guided by rehabilitative strategies.
Excellent. So if I understand your question correctly, you're asking whether a lot of the recruitment can be linked to attention, and whether attention can also be have an explanatory role in the rest of the recruitment and in rehabilitation. So in terms of, sorry, was that correct?
Yes, I'm sorry, that's right.
Okay. Sorry, I thought you said something. Definitely attention plays a role in that. Attention is hard to segregate pure attention from attention to a different sense, to attention to spatial location, to attention to. So I agree with you that is probably the role of a lot of the non-visual connectivity or feedback connectivity to V1 that may allow guiding the blind visual cortex to those functions. But executive function per se, or attention per se, has been only quite minimally linked to plasticity in primary visual cortex. At some point, the field began titrating it towards attention to touch, attention to audition, and so forth. So it's more of a fine-tuning approach of how attention to what plays a role in all those things.
But what I was trying to ask was whether a deployment of attentional control during the training of the blind could actually help guide the formation of these new connections or these strengthened connections.
So you're talking about the Prakash data per se, specifically?
Well, not just Prakash, but also the developing appropriate function in those who have enduring blindness.
Yeah, I completely agree that is likely to be the case. I wonder to what degree it also is affected by the specific compensatory strategies that the blind people are taught in different places, and what are they learning to attend and how that plays a role as well. I hope that answers the question. I'm not sure I understood.
I also have a question, thinking about it in kind of like a reverse manner, and I'm not sure you'll be able to even investigate that, but I was thinking, do you think that, I mean, people are born with different brains, and the strength of the connectivity in my brain or in your brain are different between different areas, although, you know, the main routes are there, but there are different strengths between individuals. So I'm wondering whether the people, the blind individuals who are able to improve more, maybe relying on, you know, better, stronger connections that are there to begin with. And so would you predict that if you would be able to scan them beforehand, for example, DTI, or do some, not only functional connectivity, but actually assess the wiring, the actual hardware, you would find that that would be a good prediction of the visual, I don't know, the visual acuity or the visual functional improvements. I don't know, just thoughts.
Oh yeah, yeah. The short answer is yes. Of course, that would be the optimal situation, right? To be able to a priori map the brains as early as possible if somebody's born blind, and try to see where that individual kid's strengths might be and work best with those, right? I'm not sure DTI is the right tool for that, simply not because, I mean, again, I use functional connectivity as a proxy for anatomical connectivity and for functional weights of that connectivity. Because DTI, to my knowledge, is currently still limited in allowing to test such fine-tuned parameters. So I use functional connectivity instead. But that is definitely the approach.
Yeah, but with functional connectivity, you don't, I mean, you could see that things are, you know, you could see two areas that are very highly connected, maybe because there's a lot of energy or effort investigated in this network, but it may be relying on very, let's say, thin or not very strong wiring. Whereas I'm thinking about the actual, but again, yeah, and DTI definitely is also a probabilistic approach. So there are limitations to every method.
Yeah. But again, I only use functional connectivity as a proxy for anatomical connectivity. The best situation would be to map in detail the anatomical connectivity and see what's there. I'm just not sure I have the technical capacities at this point to do it.
Yeah, definitely. It's very much hand waving, conceptual inspiration. So Ella, thank you very much for a wonderful and inspiring talk. I'd like to again thank you for joining us. And next week at the same time, which is not a regular time, but at the same time in your local area, let's say, we have Marlene Berman, again, East Coast, 8 a.m., UK, 1 p.m., main Europe, 2 p.m., and Israel, 3 p.m. next week. Marlene Berman will be with us. So thanks again.
Thank you very much. Have a great week.
You too.
Bye.
Thank you, Sharon.